Welcome to the virtual practical class on lymphatic system. In this class, we will identify the histological features of various lymphatic organs. For thymus, we will identify the cortex, medulla, vessels corpuscle, and blood capillaries. In the lymph node, we will identify the capsule, subcapsular sinus, cortex, and medulla. In the spleen, we will identify the capsule, red pulp, white pulp, and the central artery. In the tonsil, we have to identify the lymphatic nodule, the lining stratified squamous epithelium, and we have to differentiate it from the other lymphatic organs. The thymus, is situated in the superior mediastinum. It has two lobes and it is covered by capsule. The thick capsule of connective tissue covers the thymus and it sends connective tissue inside the gland, and that is called the trabeculae. Through this trabeculae, blood vessels enter into the substance of the thymus. This trabeculae divides the thymus into incomplete lobules. The darker part of the lobule is called the cortex. It contains smaller lymphocytes, and the lighter part is called the medulla. You note one of the identifying features of thymus is incomplete lobule, cortex. Separate cortex are there for the lobule, but the medulla are continuous. The medulla is light colored. This is higher magnification of the thymus, showing the trabeculae and the blood vessels. The blood vessels follow the trabeculae and divide into arterioles and capillaries. The cortex is darker and the medulla is lighter. Inside the medulla, eosinophilic substance is visible, that is the Husserl's corpuscle. There are a lot of capillaries in the junction between the cortex and medulla, and these capillaries form the blood thymic barrier. This is also lower power view of the lobule of the thymus gland, where you can see capsule, the cortex, darker color cortex, and the lighter color medulla. The arterioles are visible in the cortex, and there are lots of Husserl's corpuscle in the medulla. So one of the identifying features or the specific features for identification of the thymus is the presence of Husserl's corpuscles. The Husserl's corpuscles are nothing but the epithelioreticular cells, which are light colored cells among the dark colored T lymphocytes. The epithelioreticular cells forms the thymic corpuscle or Husserl's corpuscle. Actually, the concentric ring of degenerated epithelioreticular cells forms the Husserl's corpuscle. There is a jelly-like material inside the Husserl's corpuscle. Sometimes this corpuscle or the center of the corpuscle calcifies also. This is the lower and higher part picture of the thymus, showing the capsule, the thymic corpuscle, the epithelioreticular cells, Here is one big Hess's corpuscle in higher power. You can see the elongated epithelial cells and the central degenerated part. There are other cells also in the medulla, and there are arterioles also, and the epithelio-reticular cells which is scattered inside the medulla. There are lots of capillaries present at the junction between the medulla and the cortex. This is showing the blood capillaries of the thymus. The blood capillaries are of special nature because it is like continuous capillaries. It has endothelium, the basement membrane, and other cells also. This whole capillary is surrounded by certain other structures, macrophages, epithelio-reticular cells, and lymphocytes. On the other side also you can see one capillary, and there are lymphocytes surrounding the capillaries. This is showing the schematic diagram of the capillary wall of a thymus gland. Here is the lumen of the capillary. It's lined by the endothelium, then basal lamina. The pericytes are also there. So this is the capillary wall. Surrounding it are the perivascular connective tissue, which contains the macrophages and few connective tissue fibers. Outside it lies the epithelio-reticular layer. The epithelio-reticular cells are lying in the basement membrane, and it is surrounded by T lymphocytes. So that forms the blood thymus barrier. It doesn't allow many of the antigens to pass to the thymus. Blood thymus barrier is absent in the medulla of the thymus. Now let us come to the lymph node. This is showing the section of the lymph node and schematic representation of the section. The outer part of a lymph node is dark color and it contains lymphatic nodules. This is also called the cortex. The cortex is covered by capsule. The afferent lymphatic vessel pierces the capsule to enter the substance of the lymph node. There is a depression on one of the sides of the lymph node. It is called the hilum, through which the blood vessel enters, and the veins and the efferent lymph vessel comes out. The deep part of the lymph node is called medulla, which contains cord-like structures and sinuses, the medullary cords and medullary sinuses. This is showing part of the section of the lymph node. The outer part, the outermost part is covered by the capsule, which sends some fibers inside. Those are the trabeculae, and in the trabeculae it divides the cortex into different compartments, and each compartment contains 2 or 3 lymphatic nodules. The center of the lymphatic nodules may be dark or lighter according to the antigen challenge. These lymphatic nodules are the sites for the B lymphocytes. The inner part of the cortex, more or less nodule, is absent. This is the junction between the medulla and the cortex. This is called inner cortex or paracortex. These regions are very important because high endothelial venules are present here. The medulla contains columns of lymphatic tissue, reticular tissue, those are called the medullary cords, and in between there are spaces. Those spaces are the medullary sinuses. Medullary cords contain B lymphocytes. This is higher magnification of a lymph node showing the capsule. Just below the capsule is the subcapsular sinus. Then beneath that is the lymphatic nodule, and which contains the B lymphocytes. Deep to the lymphatic nodule is the paracortical region, which contains T lymphocytes. The germinal center of the lymphatic nodule, it contains highly packed lymphocytes, and the periphery of these germinal centers, there are special types of veins are there. Already I have mentioned high endothelial venules. See, this lumen contains some blood cells and it is lined by cuboidal epithelium. There are gaps in between the cuboidal epithelium. So lymphocytes from high endothelial venule pass through these gaps to the substance of the lymph node. High endothelial venules are absent in the spleen. The deep part of the cortex or the paracortex contains scattered lymphocytes. Macrophages are there, lots of macrophages are there. Macrophages contains big nucleus with the cytoplasm, and its main function are to phagocyte the foreign bodies or antigens. There are high endothelial lymphatic venules are there in the deep part of the cortex. You can see the high endothelial venules are lined by cuboidal epithelium, and there are gaps in between the cuboidal epithelium, and it has lot of reticular cells and reticular fibers in the deep part of the cortex. This is high magnification of lymph node showing the capsule and the subcapsular sinus containing the reticular cells, the lymphocytes, macrophages, and deep to it lies the cortex of the lymphocytes or cortex of the lymph nodes. The cortex of the lymph nodes, as you see here, contains the subcapsular sinuses, and the lymph from the subcapsular sinus pass to the medullary sinus, and which comes out as efferent lymph vessels. This is lymphatic nodule in the outer cortex containing the B cells, and these are the medullary cords which contains reticular fibers and lymphocytes. In between there are sinuses. This is the medulla of the lymph node. The medulla contains medullary cords. As I was talking, there are reticular fibers and lymphocytes and macrophages are there. In between these spaces are the medullary sinuses. This medullary sinuses receives lymph from the paracortical sinus, and those sinuses are continuous with the medullary sinus. Through that, or there is connection between the paracortical or subcapsular sinuses, trabecular sinuses, then it enters into the medullary sinuses. These are showing the medullary cord and the medullary sinuses in the deeper part of the lymph node. Here are the medullary cords and the spaces at the medullary sinuses. The spleen is lined by capsule, and the trabeculae are there, which are extensions of the connective tissue of the capsule, dividing the spleen into different compartments. There are some bluish dark blue areas. These are the white pulp, and in between the white pulps are the red pulps. This is the low magnification of the spleen, showing the trabeculae, capsule, and the trabeculae contains the blood vessels. One can see the white pulp and the red pulp. White pulp contains the lymphatic nodule and one artery. This is the lymphatic nodule of the spleen, and this is the white pulp area. In the periphery of the nodule, there is an artery. This artery is surrounded by T lymphocytes. This is called periarteriolar lymphatic sheath, or PALS. In between the white pulp is the red pulp, and one trabecule can be seen in the red pulp. This is the actual picture of spleen, showing the lymphatic nodule and the red pulp and white pulp. One can see the blood vessel eccentric position in the lymphatic nodule. This is the red pulp, which contains Billroth cords or splenic cord, which is formed by the reticular fibers, and the lymphocytes are there in between. Between the cords are the splenic sinusoids, which contains mainly RBC. This is showing the splenic circulation. The trabecular arteries divide and give central arteries, which pass through the lymphatic nodule, and it gives branches to the lymphatic nodule of the white pulp. When it comes out from the lymphatic nodule, it gives peninsular arteries, which drain into the sinusoids or splenic sinusoids. This is called closed splenic circulation. And there is open circulation also, where the blood vessel opens into the red pulp. Then from the red pulp, the blood passes in between the gaps in the splenic sinuses, and splenic sinuses ultimately form the trabecular veins. Let us see the tonsil. The palatine tonsil is present at the end of the oral cavity. Oral cavity is lined by stratified squamous non-keratinized epithelium, and there are deep to this epithelium there are lymphatic nodules. The lymphatic nodules are usually secondary lymphatic nodules in the central part will be lighter, the peripheral part is darker. The characteristic feature of the tonsil is presence of depression. Those are called tonsillar crypts. The tonsillar crypts are also lined by stratified squamous epithelium. Here is the stratified squamous epithelium and the lymph nodule. In the whole of the intestine and in the bronchial tree, there are collection of lymphocytes. In the intestine, in the small intestine, there is huge collection of lymphocytes, that is called the Pearce patch. In the appendix also, there are collection of lymphocytes. One can see under the microscope. So, in short, the identifying features of the different lymphoid organs. The thymus, lymph node, spleen, tonsil and Pearce patches. You have to see the slides and you have to differentiate one from the other organ. The capsule is there in thymus, lymph node and spleen. Tonsil and the Pearce patches, capsule is not there towards the luminal side, or remnant of capsule will be there only on the external surface of the tonsil and Pearce patch. The subcapsular sinus, it is present only in the lymph node. Subcapsular sinus is absent in the thymus, spleen, tonsil and Pearce patch. The trabeculae or extension of the capsule inside the gland are present in the thymus, lymph node, spleen. It is absent in the tonsil and Pearce patch. Lymphatic nodule. Those are the collection of lymphocytes in the thymus, lymph node, spleen, tonsil and Pearce patch. Lymphatic nodules are there in all those lymphatic organs. The lymphatic organs that can be divided into cortex and medulla. The thymus has cortex and medulla. The cortex is darker and the medulla is light colored. In case of the lymph nodes, the cortex contains lymphatic nodules. There is no definite cortex or medulla in the spleen, tonsil and Pearce patches. Medulla is present in the thymus and 4 to 5 medulla are continuous because the lobule formation is incomplete. In the lymph node, the medulla contains medullary cords and medullary sinuses. Medulla is absent in the spleen, tonsil and the Pearce patch. Hessus corpuscle are unique characteristic features of the epithelio-reticular cells of the thymus. These are the degenerated epithelio-reticular cells. It is absent in lymph node, spleen, tonsil and Pearce patch. Red pulp is the characteristic feature of the spleen. It is absent in all other lymphoid organs. White pulp is also a characteristic feature of the spleen. So, to identify the spleen, you have to identify the following features: capsule, absence of subcapsular sinus, trabecule is present, lymphatic nodule is present, and those nodules are called the white pulp. In between the lymphatic nodules are the red pulp. Surface epithelium. Surface epithelium is present only in the tonsil and Pearce patch. As tonsil lines the oral cavity, it is lined by the stratified squamous epithelium, and the Pearce patch is part of the small intestine, so it is lined by columnar cells. Crypts or depressions only present in the tonsil. Thank you. This is the end of the presentation.